you have. I want you to make a piece of art of some sort that is depicting cellular respiration. T-shirt, for example, is a piece of art depicting cellular respiration. So one of my students made this for me a couple semesters ago and got 10 points of extra credit. So you could do anything. I've had shower curtains, regular curtains, sheets, posters. Somebody made a soccer ball last semester, which was pretty cool. Aprons. I mean, whatever you want. But it needs to depict cellular respiration. So I will post to D2L a couple of examples. But here is what needs to be included on that. You need to cover in detail the process of glycolysis. So you need to show every single reactant that is involved in glycolysis. Every single time ATP is made or spent, you need to show that. You need to show what the product is of the entire glycolysis reaction. I want you to show how that feeds into the TCA cycle. And I want to see the entire cycle. So every single step of that cycle. And then how that feeds into the electron transport chain. And so I want to see all of the different complexes of the electron transport chain happening in the membrane. And most importantly, I want to see fermentation. So all 4 of these need to be shown on there. Just get creative, have fun with it. You can do it however you want.
Bless you.
And that is going to be due the day of exam 2. And if you have it with you on the day of exam 2, you can use it on the exam.
Oh, sweet.
So, if that's not incentive, that's
Right.
incentive. What is? Then again, I don't want you to memorize the steps anyway. So, but it might help you. So, you can bring that with you. If you have questions about what you can make, you can just ask me and I'll let you know if I think that that's acceptable or whatever. But the more detail you have, the more detail that you will have with you on the exam. So keep that. Yes?
So the 10 extra points would go on your exam?
On your exam. Yep.
Do you keep the extra, do you keep?
It's up to you. If you want me to keep it, I'll keep it. And if you want to take it, you can. Yep. Totally up to you. Somebody last year used like a wood Dremel and like made me 3 like wooden planks that I have hanging up in my office now, which is pretty cool. So whatever you guys want. What did you say?
I said it's a high bar they're setting. You need to lower that bar.
Well, and people have done as much as just making a poster board, which is totally fine. That's
Okay.
completely fine. Whatever you guys want to do. I know that you're studying for your exam too, but this is going to help you to
Right.
study as well, to write out all of these steps, because at no point in class are we going to write out every single step. So just keep that in mind. Okay. So the metabolic strategies. This is where we left off before. We have aerobic respiration, anaerobic respiration, right? So either using oxygen as the final electron acceptor or using some other molecule as the final electron acceptor, and fermentation, when you cut off oxygen to a species that would normally use oxygen and they have to take a different route in order to make ATP. So the whole idea is to take glucose and strip it of all of its electrons, which are chock full of energy. Then we use that energy from the catabolic reaction of breaking down glucose to form ATP, which costs energy, right? Because we're trying to add that 3rd phosphate onto ADP, and ADP is really happy just with 2 phosphates. So trying to get it to add that 1st phosphate is gonna, or that 3rd phosphate is gonna cost us. So we have to pay for that with the energy. Okay, and so here is an overview of what all of those different pathways looks like. So what you'll notice is aerobic and anaerobic respiration look very similar. The only difference is here at the end in the electron transport chain. So normally in aerobic respiration, and by the way, who carries out aerobic respiration? Who's an example? Us, right? We eat, we breathe oxygen, and that combination gives us energy. And the product of that reaction from us eating and breathing in oxygen is one, the formation of ATP, which allows us to do metabolic work, but also we breathe out two products. What do we breathe out? Yep, CO2 and H2O. So those are going to be the products of that. That being because O2 is our final electron acceptor. So when electrons are added to O2, it becomes H2O. CO2 is going to come from the TCA cycle. Okay, so there is somewhere that each of these products is going to come from. Now the difference being in anaerobic respiration, O2 is not the final electron acceptor. So water is not what is formed. And so what is formed is going to be dependent on what is being used as that final electron acceptor. So I won't ask you specifically what is formed based on anaerobic respiration because it's as diverse as the organisms who carry it out. So some organisms that maybe live in a sulfuric environment are going to use sulfate as their final electron acceptor, and so then they might make sulfuric acid. Some are going to use nitrate. They might make nitric acid. So it just depends on the organism. So I won't be asking you that, but it is important to note that you're going to have a different final product coming out of that, rather than just water. Now fermentation, like I said, this is the pathway in which the organism follows if oxygen is cut off to the organism. So it can no longer carry out the electron transport chain because it doesn't have O2 to pick up those final electrons. So it can't carry out this entire process. If it can't do that, it's got to find an alternative route in order to make ATP because regardless of whether oxygen is present or not, the organism needs to make energy. So it's just going to have to find a different route, and we'll talk about that towards the end. Any questions so far of this overview? Okay. Another thing that I want you to look at is what some of the products are that we see coming out of here. So not just the electron transport chain where we see either water or some other molecule, but maybe also of glycolysis. So no matter what, the product of glycolysis is going to be pyruvate or pyruvic acid. Don't let this trip you up. Your book's going to kind of interchangeably use pyruvate or pyruvic acid, but in chemical terms, they're the exact same thing. So either one is acceptable. And the Krebs cycle is always going to generate the same, the same products as well. So no matter what, in the Krebs cycle, a big product that's coming off of that is going to be CO2. We're also going to see some NADH coming out of these guys. We're going to see some FADH too. So remember, these are our electron carriers picking up electrons. Where are they traveling to? The electron transport chain. Okay? So no matter what, those are going to be the same products. So I want to make sure that if I ask you, what are the products of glycolysis, you can tell me what's coming out of glycolysis. Whether that means, some ATPs, some NADHs, some pyruvates, whatever, but you need to know what those products are. So as we're going through this stuff, anything that is found in red text is, or next to red text are things that I want you to know. So red is very important. So I guess maybe I shouldn't say that for this slide because I want you to know Krebs cycle. Make sure you know that. But there will be stuff in red when we go over the Krebs cycle specifically. Okay. So I think really we've covered a lot of this. We can break down aerobic respiration into 4 stages. And so those are a lot of the stages that I'm telling you to cover here, 3 out of 4. So we start out with glycolysis, where we convert glucose into pyruvate. And so all that's happening there is substrate being converted into product, right? In simple terms. Who is carrying out that reaction from substrate to product? Who's helping to speed that reaction forward? Enzymes. Excellent. So that's why we learned about enzymes. So we're going to look at some of the enzymes that are important in glycolysis. You certainly don't have to memorize any of the enzymes, but we're going to look at why they're important and what they do. And we're going to look at some of their regulation. So we can go back and directly apply that information about enzymes and how they're regulated by a product. So we have glycolysis, glucose into pyruvate. Then the TCA cycle. However, when we learn about the TCA cycle, what you will notice right off the bat is it's not pyruvate that's directly being fed into that cycle. It's actually a different reactant. So there's this little step in between glycolysis and the TCA cycle that I like to call the transition step. And the transition step is where pyruvate is converted into something that can then be fed into the TCA cycle. After the TCA cycle, which is just, again, reactant being converted into product, which is still breaking down that molecule of glucose into smaller pieces, the electrons from the TCA cycle are then going to travel to the electron transport chain. In the electron transport chain, all of those electrons that have culminated from glycolysis and from the TCA cycle are going to basically participate in a relay race. So they're going to be handed off from one electron carrier to another over and over again until they are accepted by O2. So O2 is going to pick up all of those electrons eventually after they've been handed off from carrier to carrier, and it's going to be converted into H2O. So that is a very generalized overview of what is happening in aerobic respiration. Any overarching questions so far before we get into the details? Okay. So glycolysis. This is the figure from your text. It's very small up here. So if you need to go back through this in your textbook, through every stage, by all means, I encourage you to do that. What is important about this figure, not only that it's showing you what the hell is going on, but what I love about this figure is that it's showing you the number of carbons all the way through. So the carbons are outlined in pink. And so you can directly count those carbons. We know that the chemical formula for glucose is what?
C6H12O6.
C6H12O6. Excellent. So lots and lots of electrons to be collected out of this 6-carbon molecule. But what I like about this figure is it simplifies it. So it doesn't show you all the oxygens and the hydrogens and make it more complicated. It just shows you, okay, see, we're starting with 6 carbons. By the time we get to the end of glycolysis, we have a 3-carbon molecule. So is this a catabolic or anabolic reaction that is going on in glycolysis? Catabolic, right? Glycolysis. So we are breaking a 6-carbon molecule down into a 3-carbon molecule. And how and why are we doing that? So we're doing that because the electrons are being released out of that glucose molecule and being taken to the electron transport chain. So we are stripping electrons off of the C6H12O6 and using them to harness energy for the formation of ATP. So always the end goal, ATP. Okay? So as we're going through the process of glycolysis, I just want you to pay attention to the number of carbons that we see. So it starts with 1 molecule of glucose. That means every time glycolysis begins, 1 glucose is fed in. That is not to say that the only thing we ever eat is pure glucose. That is definitely not true. We eat all kinds of sugar. And then it is converted into glucose before it's fed into glycolysis. And we will cover any of those reactions. First thing that happens, group translocation. So that 1 molecule of glucose, again, anything in red is what I want you to know. So that 1 molecule of glucose is going to be brought into the cell. So C6H12O6 brought into the cell and simultaneously modified. So remember, group translocation is anytime anything is brought into the cell and simultaneously modified. How is this modified? Let's look at our figure here. C6H12O6, or glucose, is modified into glucose 6-phosphate. So, what's happening?
It's got a phosphate.
We're adding a phosphate. So, if we have our 6 carbons, 5, 6, now we have a phosphate. And that is the process of group translocation. Now, what else do we remember about group translocation? Is it for free?
No.
No, it costs energy, right? So right away, we're spending ATP. This makes no sense because we are trying to make ATP, right? So now we're 1 ATP in the hole already. Perfect. But as my mom says before she goes gambling, you gotta spend money to make money. You gotta bet big to win big, right? So we're spending ATP because the cell knows this is gonna be an investment. So we're okay with spending it at first. Okay, the next step of importance. So there's some other stuff that happens that you guys don't need to know. Basically, the glucose is converted, the glucose 6-phosphate is converted into another form, but we don't lose any carbons. I'm receiving emails. So the glucose 6-phosphate is made into fructose 6-phosphate. Glucose and fructose are more or less the same thing. They are what we call isomers of each other. So isomers just means that things get rearranged a little bit, but they're essentially the same. So the enzyme that carries this out, which you do not have to remember, but I'm just telling you, is an isomerase. So the name of the enzyme, isomerase, makes an isomer out of the glucose 6-phosphate into fructose 6-phosphate. Now the next step from here is to go from fructose 6-phosphate. So now we're inside of the cell. So we have fructose 6-phosphate. And now it's going to be modified into fructose 1,6-bisphosphate. Big words. Not much about this that you need to know, but what you can pull from this is a little bit of information. So first, we have fructose 6-phosphate. The 6 means that the phosphate group is located on the 6th carbon of that sugar molecule. So we count our carbons and we label them just to give us an idea of what this molecule looks like if we were to look at it. Not super important. But then that is converted into fructose 1,6-bisphosphate. So what do you think is happening here? We have a 6-phosphate. Now we have 1,6-bisphosphate. What do you think of when you look at bi there? 2. So now we have a phosphate group on carbon number 1 and carbon number 6. Does that make sense? Fructose 1,6-bisphosphate. So now this 6-carbon molecule has 2 phosphates attached to it. Where do you think that phosphate comes from? ATP again. So now we're 2 ATP in the whole. So we're still spending money to make money. So at this point we are 2 ATPs in the whole. We have to keep that in mind when we're looking at our little bank account here. We're at -2 right now. And this has 2 phosphate groups added to it. Okay? Now, we'll come back to this here in a little bit, but one of the enzymes I do want you to memorize and know is the enzyme that carries out this addition of that 2nd phosphate group. And this is phosphofructokinase. So a little bit of biochemistry for you, for your reading enjoyment. Kinase, what do kinases do? Does anybody know? Any biochemists here? Where's Michael when you need him? So, good guess. Kinases add phosphate groups. Anytime you see a kinase acting, you know a phosphate is involved. So, phosphofructokinase is adding a phosphate group to a fructose. Does that help?
Yeah.
So hopefully that helps you remember the enzyme. So, phosphofructokinase. That one you need to memorize. Okay. Why we want to memorize that, I will get to in a little bit. This enzyme is really important. We call this step happening here the committed step. pardon my language, but this is where shit's getting real. Okay? That's how you can remember this. Okay? This is where we know we're going to go forward with this. We are committed. Okay?
Is it the kinase or the
We'll talk about why here in just a second. This enzyme is highly regulated that carries this out because this is the step where we're investing that second ATP. And so at this point it's like we're doubling down. We're spending a lot of money. We want to make sure we're spending it for a reason. So we're not going to spend 2 ATPs and then back out. At this point, we're 2 ATPs in the hole. We're carrying this out. We're going to finish it. We started it. We're going to finish it. Does that make sense? Okay. Wow. Lots of emails coming through Front Range right now. So ends with, as in glycolysis ends with 2 pyruvate molecules. So that's another thing I want you to absolutely know is the product that's coming out of this thing is 2 pyruvate molecules. So again, that's another thing I love about this figure is it shows us our 2 pyruvates, and you'll notice those are 3 carbon molecules. So we are splitting a 6 carbon molecule into 2 3 carbon molecules. And energy can never be created nor destroyed. Makes perfect sense. We started with 6 carbons, we end with 6 carbons. They're just split up because the carbons aren't going to go anywhere. They have to be somewhere. All right, so in the process, this is the good part. We do make 4 total ATPs. So we are making energy. But because of our initial investment of 2 ATPs, the total ATPs that could come out of this is only 2. 4 plus negative 2 is 2. So the total number of ATPs that really come out of this is only going to be 2. That's another thing that I want you to know, okay? And another product that comes out of here are NADHs. Why do we care about NADHs? What is an NADH? An electron carrier. So it's the pizza delivery guy. It's delivering electrons to the electron transport chain. So maybe we're only making 2 ATPs in this direct process, but those electrons are worth something too. So eventually they're going to pay off in the production of ATP in the electron transport chain. So that's why the NADHs are an important product as well. And you can find the NADHs in a lot of the reactions where we are losing something off of the group. So none of these steps do I want you to memorize, but what I want you to see is that now over here we have this product that is 2 3 carbon molecules. They might be identical, they might be very similar looking. We're going to keep it basic. They have 3 carbons and a phosphate on them. So if we come back to this step and go down a couple steps, we have our fructose 1,6-bisphosphate. That means, okay, we have 6 carbons and we have 2 phosphates, right? So those are even numbers. We can split this into 2. And that's going to give us 3 carbon molecules with 1 phosphate on them, right? So this whole process here is just the idea of splitting things up into those 2 3 carbon molecules that have 1 phosphate attached to them. So at this point, I'm right here. So we've split those guys up. Again, all in the process of ripping and stripping electrons off of this carbon molecule. So then what we're going to do is we're going to take that phosphate off. So we're going to take that phosphate off, and that is where we get substrate level phosphorylation that we talked about before. So we are going from diphosphoglyceric acid, nothing that you need to remember or memorize, but this is the substrate that is having its phosphates taken off of it. The phosphates are going to be added to ADP, and ADP plus a phosphate is ATP. So we get phosphorylation of ADP to make ATP. Where does that phosphate come from to participate in the phosphorylation? From a substrate that is in the glycolysis cycle or chain. Does that make sense? So important steps here where you see ATP, and here where you see ATP being made, make sure that you know that these are substrate level phosphorylations happening here, where this ATP is being made, because the phosphate is coming from a substrate that participates in glycolysis. Okay, so we're gonna skip ahead because this stuff is really not anything that I want you to know, to this step where we have a molecule called phosphoenol pyruvate, or your book at this point is calling it phosphoenol pyruvic acid. Same thing. So this is our committed step. That's eventually going to take us to our phosphoenol pyruvate. This takes us back to what we learned about enzyme regulation. And Sarah, I think it was you that said, okay, why does this matter? Why does it apply? Why does feedback inhibition matter? This is why. So because the cell is cheap, and because it's not going to spend money unless it knows it's making an investment, like it's spending money here, this whole process is highly regulated. We're not going to spend money in glycolysis unless we know that it's going to be worth it. So the product of this being pyruvate, right? Unless we know that we need pyruvate, we're not going to follow glycolysis all the way through. We're not going to invest that money unless we know that we need to. So not quite pyruvate, but phosphoenol pyruvate, which is right before pyruvate. We have this product. If we have a buildup of this product, we know this product is eventually going to be converted into pyruvate, right?
Mm-hmm.
If we have a buildup of pyruvate, why would we carry out glycolysis if we know we have enough?
Mm-hmm.
We wouldn't, right? The cell would say, I'm not spending 2 ATPs if I have plenty of this. So we see feedback inhibition at its finest participating here. Phosphoenol pyruvate is the allosteric or feedback inhibitor of phosphofructokinase. So, I'm going to just let that sink in for a minute. Catch up your notes. We're there. We're almost at the end of glycolysis. Okay. So, we have